Before we get going today, Eric and I would like to invite you all to take part in our very first question and answer session. We are going to sit down, answer your questions about aging, biology, research, translational medicine, whatever you can think of. Visit buckinstitute.org/podcast and send us in your most difficult questions, then tune in for the answers on an upcoming episode. Now, let's get on with the show. This episode is presented by Ashton Thomas Private Wealth, guiding families and institutions with clarity today and strength that endures for generations.
When you think about a worm, it seems like another world, but actually the genes that it uses to be an animal are the same. And that means that you can study a little tiny worm that lives a couple weeks and doesn't look anything like us and make a fundamental discovery that really changes the course of science.
Aging is evolving. No longer are we subject to forces beyond our understanding and control. We have charted the landscape and explored the frontiers of aging.
What was science fiction is close to becoming reality, restoring sight, repairing tissues, reviving cells, organs, and maybe even our minds.
I'm Eric Verdin, CEO of the Buck Institute.
And I'm Brianna Stubbs, a scientist here at the Buck.
On this podcast, we dive deep into geroscience, studying the intersection of aging and disease with some of the brightest scientific stars on the planet.
Join us because
we're not getting any younger, yet. Hey, Brianna.
Hey, Eric, how was your last podcast interview?
It was amazing. So I had Cynthia Kenyon on.
Cynthia Kenyon, is just such a great in the field. The very first person, I believe, to show that aging could be regulated by our genes.
The second one, actually, Tom Johnson was the first one in Colorado. But nonetheless, she was the first one that actually identified a pathway that controlled the aging process, an insulin signaling pathway.
I feel like with so many things in aging, it comes back to metabolism and insulin signaling being one of those most important central regulators of the way we age.
Yeah, what she certainly contributed is an amazing, completely new view of aging. When I was at UCSF, it just was a bomb that exploded in the whole field of aging in terms of the significance of what it means. And the chance to talk to her and hear how this all happened was really unique.
And I know she uses the C. elegans worm model, right? Well, I use humans as a clinical model, so I'd be interested to see how what she's doing relates to the clinical work that I do here at Buck.
It relates actually amazingly, and this is why, I've been a big defender of all of these animal models. They're important because what we've learned applies not only in the C. elegans worm, but every species, including humans.
I really can't wait to hear this one.
Enjoy. Today I have the immense pleasure of speaking with someone who has truly shaped the field of aging science, Dr. Cynthia Kenyon. Cynthia is a vice president at Calico, a member of the National Academy of Science. She's one of the true pioneers of what I call the modern science of aging. Now, Cynthia, you may not know this, but your early work at UCSF, where I had the privilege of being one of your colleagues nearly 30 years ago, was actually one of the key reasons I decided to pursue aging research myself. So I want to start by going back to these early days. Can you describe for our audience what was the field of aging research like when you started?
Well, it was very, very, very different at the time. The general idea was that aging just happened. You just wear out. People knew that different species age at different rates, and different species differ by their genes. And if you think about it, that means that there have to be genes for aging. A lot of people didn't really think about that. But they also thought that if there were genes for aging, there would be so many. There would be different genes for the skin and other genes for the heart. Maybe there would be lots of genes with little effects. So even if there were genes for aging, you'd never find them. And so there was a basic idea that, there's nothing you could do about it.
Can you actually walk us through that first pivotal experiment? What was the spark that led to the identification of these mutations?
Well, it's interesting. It's a very interesting story. Like I said, the basic paradigm was that there wouldn't be genes for aging. And yet, so I worked on a little tiny animal called C. elegans, which is a roundworm. It's about the size of a comma in a sentence, very small. You can just barely see it with your eye, and you can see it with a microscope. But they age really quickly and die in just about 2 weeks. So actually, it's a nice little organism if you want to study aging. So before me, a few other scientists had asked whether you could change genes and extend the lifespan of these animals. So the basic idea here is that if there are genes for aging, and there have to be, because different species age at different rates, if there are genes for aging, if there's, maybe there's some genes that have a big effect, maybe, then if you change them, well, you could extend the lifespan of an animal. I was previously involved in studies with C. elegans that showed that the ability of animals to generate body patterns like arms and legs and things like that in certain places as they developed was due to the presence of some universal master control genes that operated in all animals to make body patterns. Now, this is not something that you would think would exist. You could change these genes and a fruit fly would develop an extra set of wings, or the antenna would turn into legs. So you got these huge changes in body pattern by making single changes in important master control genes. So my idea was that there were master control genes for aging. Why not? All animals age, they do so at different rates. Maybe there's something like a thermostat for setting the temperature, except it's kind of a thermostat for aging. So basically you could turn it one way and shorten lifespan and the other way and lengthen lifespan. And the thermostat would work the same way in all species, but they would be set differently in different species. So some animals would age quickly, some animals would age slowly. So I came into this field with the idea that there really might be master control genes for aging, individual genes that if you change them, you could get a big effect. And we found miraculously, it wasn't miraculous, but we were very lucky to find early in our exploration single mutations in single genes that doubled the lifespan of the animal and didn't affect its feeding. They were not calorically restricted, and it didn't affect their fertility very much at all, like barely at all. So, and then we showed even that you could change these same genes when the animals were already adults and when they had already finished reproduction, and you could still get them to live longer. So that was a big deal, because that said there were individual genes that controlled the rate of aging, that aging was plastic, it wasn't fixed in stone, and it could be changed, which was amazing. And then later on, work from my lab and other labs showed that the genes that we had found in my lab to affect aging encoded very familiar kinds of molecular proteins that affect hormone signaling and the responses that cells had to hormones. So basically, what we'd found was that molecular biology that we already knew about and understood pretty well was controlling aging. So suddenly you could study aging and you could use the same tools that you'd already been studying it. One of the genes that we found that controlled aging was a gene called a transcription factor, which is essentially a gene regulator, which turns different genes in the DNA up or down. That gene was needed for the lifespan increase in these animals. So it was a very much of a program, if you will, like a computer program, where you have regulatory genes, control genes, and you have a hierarchy of commands. You have master regulators, and then you have the effectors. It was amazing, and it was controlling aging, which everyone thought just happened. It was really boring.
And equally remarkable was the fact that you not only found one gene, but you found several, and these were organized in a pathway that normally works together in the cell, the insulin signaling pathway.
Right.
And so from there, others were able to identify the same pathway in other model organisms, correct?
That's right. I mean, actually, it's very interesting. We published our paper in December, and the March before then, one of the leaders of the field of aging had written an article in Nature, which is a very prestigious magazine, saying there wouldn't be genes that had big effects on aging. There couldn't be. And this was a complete paradigm shift. And then, in fact, that same scientist studied fruit flies, and she she asked, okay, there are gene changes that affect this same hormone pathway in the fly. I wonder if they live longer. And they did. And then other people asked the same questions in the mouse, and the mice live longer. That's important because mice are mammals like we are. So that's important because it says that from evolutionary theory, it says that the ability of this hormone signaling pathway to control the rate of aging is ancient. It had to be up and running in a common ancestor, evolutionary ancestor that gave rise to the worm and these fruit flies and mice. And that same common precursor also gave rise to humans. So, and we know that the insulin signaling pathway is active in humans, but we don't know as if it controls aging. And we think probably it does, but we don't know that for sure.
I agree with you. And in some way, you know about the ITP, the intervention testing program that the NIH runs, which actually has identified drugs that increase lifespan in mice. And there's a remarkable number of drugs that target insulin and glucose, sugar metabolism in those, including acarbose, SGLT2 inhibitors. And of course, now we are hearing a lot about Ozempic and the GLP-1 agonists, which are emerging as potential anti-aging drugs. So this must be an incredible validation for you of all of that work.
It is. So basically, I think it's important to get the general concept, and it's not intuitively obvious, really. So basically, these hormones, the hormones that we identified with others as controlling aging, were hormones, the hormones insulin, and another one called IGF-1, which stands for insulin-like growth factor number 1. And I know you've all heard of insulin because if you don't have enough of it, you get diabetes, or if you're resistant to it, you also get diabetes. So that's very important. And that's associated with having a lot of sugar in your body, type 2 diabetes anyway. Those genes are involved in sugar metabolism and allowing your body to take up sugar after you eat a meal. IGF-1 is involved in promoting growth. But they're very closely related pathways. And basically, during a normal life, in a happy situation, you have enough to eat, you're eating, you're growing well, you reproduce, your children grow, everything is fine. And what the work on these worms showed was that if you slightly inhibit the pathway, so it doesn't work quite as well, it works, but not quite as well, what it does is it gives the body a danger signal. It says, uh-oh, maybe there's not enough food out there, or maybe there's some other bad condition like, I don't know, temperature, irradiation. There's other conditions that will do it also. And what the animal does is it rolls out a very large kind of system-wide, pervasive resiliency response. So suddenly the same animal is protected against a lot of different stresses. It makes the proteins that it makes fold better. They last longer. They're more resistant to, oxidative damage, reactive oxygen species, all these horrible things that you've heard about. So basically, when we extended the lifespan of these worms, we didn't really know what we were doing. We just looked for gene changes. The animal did it all by itself. All we had to do was change one gene, but the animal sort of knew how to do it. What it was really doing was to activate this danger system and trigger a response to a danger that really didn't exist. It thought it existed, but everything really was fine. And so out came this response, and consequently, the animal could live twice as long. So that's really important, because that's why inhibiting it a little bit can have this beneficial effect. If you inhibit it completely, you die. We need insulin. We need IGF-1. When you eat a meal, your level of glucose or sugar rises, and that causes insulin to increase, which causes the sugar to be taken up into your cells. If there's too much sugar, then the cells say, hey, give me a break. And they sort of close the door and they become resistant to insulin. And that's what causes type 2 diabetes. Okay, so basically one of these drugs, acarbose, prevents the body from taking up glucose. So you keep your glucose levels low and the animals live longer, the mice live longer. SGLT2, which is a diabetes, or inhibitors of that, which is a diabetes drug, so normally what happens when you eat sugar, your body wants to keep it inside you. It thinks it needs it. So it goes into the kidneys, and the kidneys say, oh, no, you're sugar. You don't go out. You stay here. So it has a special protein that keeps it in. And this keeps it from being just excreted in the urine. And what this drug does is it blocks that mechanism. So now when you eat meals that have sugar in them, the sugar leaves your body through urination. And so the level of sugar in your body is lower. So you have lower levels of insulin. And as a consequence, at least if you're a mouse, there are big benefits. And actually, even in humans, there are a lot of benefits of SGLT2 inhibitors. That it benefits diabetes, but other conditions as well. And also famously, so do these GLP-1 receptor agonists like Ozempic and Wegovy. Those just cause you to eat less. But actually, you also sort of change your taste preferences, apparently, so that you don't really want all that sweet food. And again, as a consequence, you're protected against diabetes, but a lot of other things also, like fatty liver disease, cardiac problems. There's emerging evidence of neurodegeneration and addictions, all sorts of the biology isn't really completely known, but there's a lot of things get better. And it's all probably part of this same system that I was talking about, how having too much sugar is not good for you.
There's a lot of discussion right now in terms of research and what it should focus on. And there's a lot of interest in translational research that is bringing basic discoveries to humans. But as the head of the buck, I've always made the case that the true importance lies in making the early discoveries, such as you did, because it essentially opened up the gate to really discoveries in humans. So can you speak about the importance of basic research in model systems such as the worm or mice or fruit flies, which appear to be so different from us at first sight, but when you look at the core mechanism, the core pathways that regulate aging, they're actually conserved all the way from the little worm to humans. Can you speak about this?
I can. This is an incredibly fundamental question, and it's a little bit counterintuitive. When you think about a worm, it seems about as different as a person as it could get. I mean, people, when they even see other kinds of people, they think, oh, they're so different from each other. People against people. But against a worm, it seems like another world. But actually, the fact of the matter is that in order for an animal to be alive and go about its daily business, it has to have a lot of the same things that a person has. It has to have a functional intestine, functional muscles, neurotransmitters. It has to have a functional nervous system. And it turns out that the genes that it uses to be an animal are the same as the genes that we use. Even though we think they're so different, they're actually much more similar than they are different. And that means that you can study a little tiny worm that lives a couple weeks and doesn't look anything like us and make a fundamental discovery that really changes the course of science. People are all over the world are trying to figure out how to slow down aging in people. But more importantly, keep us, functional and happy as we're getting old, healthy as we're getting older. I think now we can see that it's possible. And the reason was because of curiosity-driven basic science, really.
Yeah, and so studying this little worm has a number of really key advantages. First, the research is much cheaper. It lives 21 days, and so it allows really a complete manipulation of the genetic code and allows us to do many things way faster. And so they are really thought as an accelerator of the whole research enterprise. And I think we all know, that C. elegans, the little worm, helped us to make so many fundamental discoveries that are making their way into medicines today. So I think it's a really strong argument from my point of view for the support of basic research and the enthusiasm that we have have here at the Buck and many other places in studying these organisms. So that's, in my opinion, your story is one of the best example of the power of these model systems. So this is great. This is what happened then. And without exaggerating, I think it really ushered in modern aging research. And we are now, 25, 30 years later, seeing all of these discoveries moving into humans. So a few years ago, I don't remember, I think it's about 10 years ago, you made a pretty gutsy transition. That is, you left the temple of academia to go to the dark side and work for industry. At least this is how many academics refer to it. Can you tell us, and I was really admirative when you made that transition. I would love to hear, after making such a fundamental observation, you made a big move to Calico, which is now one of the alphabet company associated with Google. Why did you make that transition?
When I started out as a young professor and even as a postdoc in my training, I just really wanted to understand how body patterns were made. Why the head was different from the rest of the body, where the limbs went. And so I studied this and we learned a lot about it. But it never made me really want to go into industry or start a company. I mean, I did think it would be nice if people could fly, if we could grow wings. This occurred to me, but that was a little bit ahead of its time, still is. But anyway, then we made our discovery about worms. And I sat there and I looked at these worms. And it was at a time in their life when the normal worms were about 2 weeks old and they were almost all dead. And the ones that weren't looked like they were on their deathbed. And then right next to it was another one of these little worm homes that contained a long-lived mutant. And those worms were just moving around, looking young. And my hairs stand up. They still stand up. They're standing up right now every time I think about that, because they should have been dead. The fact is they should have been dead, but they weren't dead. They were alive and they were still young. And I looked at that, and here's what I thought. Number one, gee, I feel sorry for the old worms that are all dying. I still feel sorry for them. Then I thought, oh my God, that's going to happen to me. Then I thought, I want to be those other worms. And then I thought, oh, I want to start a company. So like within minutes, I wanted to start a company. And then, and I think about the year 2013 or so, I saw the magazine. Some people said to me, Cynthia, have you seen Time magazine? And I said, no. And they said, well, look at it. It says something like, can Google stop aging? Well, I loved Google. I thought Google was just the coolest thing, smartest people, just a great company, just the state-of-the-art computation and intelligence and ingenuity. And they were going to start a company that was going to focus on aging. And the CEO was going to be Art Levinson, who was an absolute hero of the biology world. He was a leader at one of the very best and earliest biotech companies called Genentech. And he was a legendary. And he was going to be the CEO of this company, and they were going to study aging. And I thought, gee, I could go to this company and I could recruit like super great talent, really smart people. They would all be working on aging. And it would just be a thrill. And I could contribute because I actually know a lot of details about the molecular biology of aging. And by that time, it was known that you could extend lifespan not just by changing this insulin IGF-1 system. You could also do it, or you could make animals a lot healthier if you put young blood into an old animal. For example, if you cleared, there's certain cells called senescent cells, which accumulate with age, and those are very highly inflammatory cells. And Jude Campidi's lab at the Buck had already showed that if you clear those cells, if you kill them, mice are way healthier. And and they have a longer mean lifespan. So there are all sorts of ways that you could slow down aging. It wasn't just one way, many different ways. So I just thought, oh my God, this is so great. And I don't think it was the dark side. I think it was bursting through into the light, to actually do something that could help humans, but which isn't to minimize basic research. It would never have happened without basic research. So I just thought, oh my God, something's going to work. I'm just going to have this great time and try to like figure it out, help figure out how to bring it to humans. And it wasn't just me. I knew that there were many people working on it, but it was the best way I could think of to sort of speed it up.
What has that journey been like for you, sort of moving into a translational, mission-driven organization focused on aging?
Oh, it's great. So first of all, Calico is a very interesting company because it's basic research still plus translational work, in other words, drug development. That's the first thing. The second thing is Calico doesn't only try to slow down aging. They also make drugs for age-related diseases like cancer and other diseases. So it's kind of it's a company that does many different things, which is really wonderful because all of these different units sort of synergize and make a very rich environment for identifying translational targets for aging and trying to target them with drugs to cure diseases and hopefully slow down aging while we're at it. It's really a thrill. I love it.
The field of aging in general, I think, as I see it, is divided into two broad tendencies. One is the rigorous sort of biotech approach, sort of Altos and Calico and BioAge, who are really looking for FDA-approved drug. And then there's, in parallel to this, a whole world of a proliferating world of supplements, of people taking the situation in their own hands. And I think I know we've had these discussions in the past. I would love to hear your thoughts on what these two worlds mean, the danger of one versus the other, and what is the best way that we could actually bring all of this knowledge to humans and to the public.
Yeah, this is a great question. I mean, there are all sorts of either drugs that are off patent and are easy to get, like rapamycin, metformin. There are probiotics. There's compounds found in wine called resveratrol. There's something called taurine. There are all these different precursors of NAD, all of which in studies in the laboratory have been shown to have benefits on the biology of aging in animals. And a lot of those are very cheap and easy to get, and people take them. But the problem with it is that they don't know. There's two things they don't know. One thing they don't know is whether there are going to be side effects if you keep taking something for a long time. And the other thing they don't know is if they're really going to work. And the only way to really know that is to do a large, what we would call in the world of drug development in a pharmaceutical company, a phase 3 trial, a large trial, the kind that were done for Ozempic, for example, for these GLP-1 receptor agonists, for example. Very large trials where you have lots of people and you have one group that takes a placebo, doesn't really get the drug, and the other ones that do get the drug or the intervention. Then you compare how they're doing. And the problem with that is what people in industry are doing. And it's not what people in the supplement world are doing. And the reason is that it costs a lot of money. It can cost between $500 million and even $1 billion to do one of these trials. It can cost a lot of money. And in order to do that, you have to spend the money up front, and then your trial will probably fail because most trials fail, but occasionally one doesn't fail. And then you have to sell it in order to forget making a profit for a while. You have to actually just get your expenses back. Then you can make a profit. And if your aspirin is something, for example, that has been shown to extend the lifespan of the mice a little bit, believe it or not, aspirin. So what if you showed that aspirin, increases the lifespan of people? What are you going to do? Try to compete with Bayer and make aspirin? It's just not going to happen. You can't get your money back. So I think that we really need something that I like to call the World Healthspan Organization, kind of like the World Health Organization, except for healthspan, which means how healthy you stay as you age, where lots of governments and nonprofits all chip in all around the world, and they design, carry out, and fund these studies so that we can find out if some of these really are beneficial. And there's a huge benefit to that. If we figure it out, then people can take them, and everyone can take them. They're cheap. That's what these things don't. I mean, that's the upside, is they're cheap. So you could give them to very poor people in all countries, to anybody, rich people, poor people, and they would make you more resilient. They would make you probably more disease resistant and just increase your health as you age. And actually, there's a real need for it now because, we have this demographic inversion that you've all heard about where there's more old people than young people. So it's really important to keep these, or a growing number, a growing proportion. So it's really important to keep these, aging people productive and healthy for a longer time, just economically, to say nothing of the happiness. So I really think this is a great thing, but it's a real problem. Meanwhile, people are just taking all sorts of things and combinations, and they don't know.
I
So
agree. It seems that there are a lot of low-hanging fruits that are not being tested rigorously because of pure economic reasons, and that this is one area where governments, with all the benefits that the government would accrue from life-enhancing drugs or supplements to bring to the population. So this is love your idea of having or creating some kind of consortium where all of humanity will participate. So this is a really cool idea. So what do you do personally to stay healthy and maximize your longevity? What's your daily? I'm not going to ask you about your daily routine, but anything specific that you want to share with our audience in terms of what you think is important to focus on?
well, all right. So what you always hear is diet, exercise, sleep, and social connection. Those are the things you hear about all the time, and I think they're probably all true. So diet is interesting because you have to make a choice. I know that we don't know what the best diet is for sure, but it doesn't matter. You have to eat something, so you have to just make your best guess. And we did an experiment years ago where we gave our little C. elegans worms some sugar, and which is, remember, that's what insulin responds to. And so that's something that would affect our lifespan circuitry that we discovered in the worm. Anyway, it turns out if you give them sugar, they live shorter. And we did some experiments and we found out that what was happening is that instead of turning this pathway down a little bit, which could double its lifespan, we were turning it up. So we were doing the same thing that would happen if you ate a diet that's very high in sugar, which makes you insulin resistant and gives you all these diseases and shortens your lifespan. So the day we got that result, I had heard about this diet called the Atkins diet. Someone had told me about it a few months earlier. So I remembered Atkins. So I went to the bookstore and I got the book on the Atkins diet. And then, of course, I got books on the South Beach diet and all these other diets, Mediterranean diets of all sorts. And I started eating that way. So basically what I did was I stopped eating food that would turn into sugar quickly. That means food that has what they call a low glycemic index. So I started eating that. So it means no potatoes, no white food, essentially, no rice, no potatoes, no bread. I stopped eating all that. Lots of salads, lots of nuts, some fruit, lots and lots of vegetables, eggs, some poultry, fish, sauces of all sorts. And I've been doing that since 2002. So it's actually quite extreme. And I really do it. People are always laughing at me because I will eat pizza, but I just eat the top and I throw away the crust. It's kind of embarrassing. But anyway, that's what I do. I also exercise, especially lifting weights and doing aerobic exercise. Those are the two main things that I do.
Very cool. I think what you're describing is, in essence, a ketogenic diet.
It's not ketogenic. I'm not extreme enough that I'm in ketosis. I'm just not eating a kind of, what is it called? Ultra processed. I mean, it's like the total opposite of ultra processed food is. It's a simple kind of but I'm not in ketosis. And I know what that feels like because when I started, I was in ketosis. I got these little strips, you can test your urine. And I know what that feels like. It's like your urine is like a fire hydrant. That's the way it is. But I'm not there. However, there are ketogenic diets, and apparently they're pretty good for you. But I don't really know that, just from studies that have been done.
What do you think is going to be normal for us in 10 years in terms of maximizing our aging? If you had to tell us, what are we all going to be doing 10 years from now?
Well, it's pretty simple. I think we'll still be trying to eat well. Hopefully, we'll have a better idea of what really is the best food to eat. We'll trying to be getting enough sleep, getting enough exercise, having good social connections. We'll still do that. And maybe we'll be taking something. Maybe there'll be something that we can eat or inject or something that will actually, intervene in some of these genetic control circuits for aging, the same circuits that make different species age at different rates. Maybe we'll make our dogs live longer, maybe that will be routine. There are companies trying to do that actually, and that that could happen.
One idea of these companies is that they're going to allow us to test some of these drugs in some of our closest relatives, our pets. And I know you, we all know about Loyal, correct?
Yeah, there's a company, Loyal, and it's run by Celine Halua, and they're testing. So it turns out that large dogs have very, very high levels of IGF-1. That's this hormone that we said can speed up aging. And large dogs have very short lifespans. They age very quickly. And so she has a bunch of different interventions that should lower the level of IGF-1 in these dogs, and that people are bringing their dogs in. They're in trials right now. And hopefully the animals will age more gracefully, maybe live longer, but have fewer diseases, be healthier as they age. It should be the case. Actually, there are mutant dogs that have gene changes that lower the level of IGF-1. Those dogs are small and they live very long. They can live to be like 18 years old, whereas these big Great Danes, for example, only live to be 7 years old. So it's really quite remarkable. And it's all apparently largely IGF-1 based. So that'll be interesting. And it'll also, I don't know what it would say about people, but it will be very interesting. You know, if it's true in dogs, it just makes it even more exciting to think about it in people.
I agree. So from the worm to the dog to humans, I think that's the pathway that we all hope will come to realize, and for the benefit of humanity at the end. Thank you so much for joining me today. I think your work continues to inspire generations of young scientists, and as we've heard, holds enormous promise for the future of human healthy aging. And I'm truly grateful for the chance to share your story, your science, and your vision with our audience. And I can't wait to see what the next decade will bring from Calico, from your lab, and from this extraordinary field that you helped to create.
Well, thank you so much, and thank you for having me. It was great to see you.
My pleasure. Same here.
Thank you so much for listening. Please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts.
We're Not Getting Any Younger Yet is produced by Vital Mind Media. The Buck Institute's very own Robin Snyder is the executive producer. Wellington Bowler is right next to us here directing the recordings, and the esteemed Sharif Azad weaves the show together for you.
If you're listening to this podcast, you know that there has never been a more exciting time in research on aging. Discoveries in the labs are moving into the clinic to help us all live better longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to learn more and to donate. This episode was sponsored by Ashton Thomas Private Wealth, where discipline shapes vision and vision builds legacy. Learn more at ashtonthomaspw.com.
Investment advisory services are provided by Ashton Thomas Private Wealth, LLC, and Ashton Thomas Securities, LLC, SEC-registered investment advisors. Securities are offered through Ashton Thomas Securities, LLC, a registered broker-dealer and member of FINRA SIPC.